My dear students, in this class, let us learn about the structure of lysosomes. Within the cell, there are various types of cell organelles. Some cell organelles are responsible for constructive activities, whereas some other cell organelles are responsible for destructive activities. Some cell organelles perform catabolic function, some other cell organelles perform anabolic functions. So, there are certain cell organelles like mitochondria which are responsible for the process of respiration, and there are some other cell organelles which are responsible for packaging and processing of different products. For example, if you take endoplasmic reticulum, ribosomes, and Golgi complex, all these cell organelles they are actively involved in the synthesis of certain cellular products. Whereas lysosomes are very unique in one particular aspect that they are destructive in nature. So lysosomes are not constructive in nature, lysosomes are destructive in nature. Lysosomes are responsible for the lysis of own cell. Lysis means breakdown. That means the entire cell will get dissolved, and sometimes the cell organelles within the cell may get dissolved by the action of lysosomes. Because of this lysis action, these cell organelles are called lysosomes, and the term lysosome was coined by Christian D. Dewey. All animal cells contain lysosomes except mature red blood cells, and these are unique cell organelles containing acid hydrolases. Hydrolases are responsible for the process of digestion, and we know pretty well that all the digestive enzymes are aptly called hydrolases collectively because the process of digestion is chemically hydrolysis. So, because lysosomes contain acid hydrolases, they can digest proteins, carbohydrates, lipids, some toxins like that. So, it is responsible for the breakdown of solid material or liquid material. Hence, these cell organelles are called lysosomes. And these are often associated with the cells of secretory and excretory nature because secretion and excretion require breakdown or lysis or the conversion of one material into another material, and all these things can be done by the lysosomes with the help of this set of enzymes called acid hydrolases. And lysosomes are called suicidal bags of cells because one cell organelles will be destroyed, one cell organelles will be dissolved by these acid hydrolases during the process of starvation. These are called suicidal bags of cells. Lysosomes are responsible for lysis of the cell. They contain destructive enzymes. Lysosomes were previously referred to as perinuclear dense bodies, and lysosomes have got dual origin because they may originate from either endoplasmic reticulum or Golgi complex. And these are the vesicles. That means lysosomes are the vesicles filled with nearly 40 types of hydrolases. And you remember all these hydrolases are involved in the activity of dissolution. That means in the breakdown of various chemical products like carbohydrates, proteins, lipids, etc. Enzymes found in lysosomes are proteases that digest proteins, nucleases that digest nucleic acids, glycosidases, lipases, phospholipases, phosphatases, and sulfatases. Lysosomes exhibit polymorphism. That means they are found in different forms. And depending on the functional status of the cell, different types of lysosomes may be found within the cell. There are primary lysosomes, secondary lysosomes, residual bodies, and autophagic vesicles. Because there are nearly four functional stages of a cell, and cells, depending on the functional status, contain four types of lysosomes, namely primary lysosomes, secondary lysosomes, residual bodies, and autophagic vesicles. So here in this slide you can find the lysosome that is fusing with the phagosome. We will learn about what is phagosome and what is phagolysosome in the subsequent slides. But here you can see clearly the structure of lysosome which is going to fuse with the phagosome. So I told that there are four types of lysosomes, namely primary lysosomes, secondary lysosomes, residual bodies, and autophagic vesicles or autophagic vacuoles. And primary lysosomes are the vesicles filled with digestive enzymes. That means these are the vesicles filled up with hydrolases. And these primary lysosomes are going to fuse with either phagosome or pinosome. And because these primary lysosomes contain all these hydrolysis enzymes. These are also called storage granules. The term storage granules is specifically applied to primary lysosomes. Let me repeat, primary lysosomes are nothing but the vesicles filled up with various digestive enzymes, and these are going to fuse with either phagosome or pinosome. And primary lysosomes, because they store various enzymes, these are also called storage granules. Secondary lysosomes are formed by the fusion of primary lysosome with phagosome. Primary lysosomes fused with phagosome or pinosome are called secondary lysosomes. That means primary lysosome plus phagosome or primary lysosome plus pinosome is collectively called secondary lysosome. That means these secondary lysosomes are made up of two parts. One is lysosome and another one is phagosome or pinosome. What is phagosome means? We know the definition of phagocytosis. Solid intake is called phagocytosis. That means if the cell ingests any solid particle, that is called phagocytosis, and such cells are called phagocytes. And WBC are the best example for this. And cell drinking is called pinocytosis. That means when the cell ingests a liquid particle, that is called pinocytosis. So whenever the cell ingests solid particle and it is bound by, this solid particle is bound by certain membranous vesicle, and this membranous vesicle that includes or that ingests, that engulfs the solid particle is called phagosome, and the vesicle that includes the liquid particle is called pinosome. So phagosome contains solid food particle, whereas pinosome contains liquid material. And if these phagosomes or pinosomes are fused with primary lysosomes, then it is called secondary lysosome. After the formation of secondary lysosome, the primary lysosomal contents in the form of hydrolysis will be just released into the phagosome particle. That means into the solid food particle or pinosome ingredients. So like that liquid food particle or solid food particle will be simply dissolved and digested by the release of this hydrolysis. So that is about the process of digestion that is being carried out by these lysosomes. Residual bodies. That means I told that the primary lysosome is going to fuse with either phagosome or pinosome. Later it becomes a secondary lysosome, and in this secondary lysosome, the lysosomal part will release certain enzymes onto the solid particle or liquid particle, thereby catalyzing the process of digestion. But after the process of digestion is over, still there are certain left out particles which are not acted upon by these digestive enzymes. That means these are called non-digestive material. So in secondary lysosomes, solid or liquid material is undoubtedly digested, but still secondary lysosomes are left with certain undigested waste materials. And the secondary lysosomes that are left with undigested waste debris or waste material are called residual bodies. That means residual bodies contain only the undigested waste material. Primary lysosomes contain acid hydrolysis, they are called storage granules. Secondary lysosomes are formed due to the fusion of phagosome or pinosome with primary lysosome. And residual bodies are those lysosomes which contain only undigested waste material. And autophagic vacuoles, of course, these are of course different set of primary or secondary or tertiary, like that lysosomes, of course, and these are totally different from this primary or any other type of lysosome. Whereas because primary lysosomes are containing acid hydrolysis, secondary lysosomes are responsible for the process of digestion by fusing with either phagosome or pinosome. Whereas autophagic vacuoles, they are not digesting any extra material, extracellular material, but they are digesting the own cell organelles. So such lysosomes, such types of lysosomes which are digesting the intracellular material, which are digesting the cell organelles within the cell, they are called autophagic vacuoles. And these are formed when the cell is not properly fed. That means autophagic vacuoles are formed only during starvation, and they are actively involved in the process of self-digestion or self-lysis or autolysis. And worn out cells also exhibit autophagy. And these autophagic vacuoles, of course, these are actively involved in the digestion of own cell particles are own cell organelles. Autophagy is nothing but self-destruction, and the own cell organelles are thus digested. And sometimes during the process of menstruation, the uterine epithelium will be simply digested. It will be peeled out and it will be digested, and it is subjected to some sort of lysis. And you can also say that this is the best example for autolysis. This is the best example for self-destruction. Like that, these are the types of lysosomes which are actively involved in the process of autolysis. Thus, coming to the functions, of course, I made a different video regarding the functions, so you can also watch that video. Just let me make the summary of this entire presentation. Lysosomes are the cell organelles that are actively involved in the process of destruction. These are the destructive cell organelles, and they contain acid hydrolases, and the term lysosome was coined by Christian Dudoué, and they're responsible for either excretion or secretion or digestion or autolysis, like that. And there are different types of lysosomes like primary lysosomes, secondary lysosomes, residual lysosomes, and autophagic vacuoles. Primary lysosomes contain acid hydrolases and they are called storage granules, whereas secondary lysosomes are formed due to the fusion of primary lysosomes with either phagosome or pinosome. And the residual bodies are those lysosomes which contain undigested waste material, undigested cellular debris. And whereas autophagic vacuoles are responsible for the digesting the own cell organelles during the starvation period. And during menstruation period, the uterine epithelium will be simply lysed or dissolved or digested with the help of this autolysis. So coming to the functions, let me give you a bird's eye view regarding the function. Extracellular digestion, intracellular digestion, autolysis, fertilization are the functions of the lysosomes, because we know pretty well that the spermatozoa contains acrosome, which contain certain enzymes that digest the egg membrane, and this involves actively the action of lysosomes. You know that there is one enzyme called hyaluronidase within the acrosome. Of course, all these enzymatic secretions and dissolution of that egg membrane during the process of fertilization is achieved or is accomplished by the collective action of lysosomes. So this is about the structure of lysosomes in specific and function in general. Thank you very much.